Hi everyone, I'm Santanu Rathod, and today I'll be presenting ContextFlow. So our task is that of trajectory inference from spatial omics data, which unlike single cell transcriptomics, retains the spatial context, thus enabling for more granular downstream analysis. And the objective is essentially to infer the temporal dynamics that best explain the snapshot kind data that we usually have in these settings, as seen on the figure here. The difficulty, however, as with any other omic profiling technology, is that of destructive measurement. So you can't really track cellular states over time. The moment you measure the cell state, the cell dies. So mathematically, you can formulate this as if you have time-varying probability distributions, and how do you characterize how these distributions are evolving over time, and every cell then can be represented as a sample from this time-varying distribution. And that's precisely what all continuous generative modeling is all about, which is to characterize the probability path that transforms a tractable source distribution to a target distribution. Flow matching is a recent addition to this line of thought, where what happens is instead of SD formulation that you have in diffusion modeling, you have a ODE formulation, and it's simulation free compared to continuous normalizing flows or ODE methods, and it's more elegant and efficient compared to diffusion modeling. Without going into much details, what it all boils down to is minimizing a regression loss, as seen in the figure here, and you essentially train something called as a neural network velocity field on conditional path. A conditional path is basically a proxy for rate of change of how a random variable is transforming some source into some target distribution. And usually you calculate this conditional path by coupling cells of consecutive slices. Now you can couple in all kinds of ways. You can do random coupling, you can come up with your own policy, but empirically it has been found that optimal transport couplings performs best. And it also has good physical interpretation in terms of minimizing the work done that's required to change a set of samples into another set of samples. However, these OT couplings suffer from a limitation in the context of spatial omics, because even though these optimal transport couplings can be statistically optimal, yet they can be biologically implausible, as seen in the figure here, where what you have is two consecutive omic slices, and every line here corresponds to a coupling that has been sampled from a transport plan. And these couplings are interesting because they are coupling between cell types that serve completely different functionality. So for example, you have an immature MSN cell coupled with an immature CMPN cell, and there's no way that MSN cell is going to transition into a CMPN cell at any point in time. But the synchron algorithm using which the optimal transport is iterated on doesn't know this. So we need to incorporate this, we need to constrain the generation and utilize domain knowledge. And the reason why we need to constrain the generation is because the flow matching model is ultimately trained on these couplings, and if you're giving it false conditional paths, eventually your generation is also going to be implausible. And that's essentially the essence of context flow. Mathematically speaking, you can have several probability paths that minimize the log likelihood that explain the data, but you do not want any probability path. You want the path that best explains or respects the underlying biology. And that's exactly what we do in context flow by incorporating prior domain knowledge. And before going on to what kind of domain knowledge should we incorporate, we should ask ourselves, what does biology tell us in the context of spatial omics? Well, it tells us that if you have two cells that are physically closer in close proximity to one another, then it's more likely that they are going to transition to a similar cell state than if they are further apart. However, in spatial omics, you do not really have a global frame of reference. So instead, what we do is we take mean neighborhood expression as a proxy for how close the cells are. Similarly, the cells do not exist in isolation. They are communicating with their neighborhood. And so it's also more likely that if two cells have similar communication patterns in terms of the ligand receptor proteins that they're expressing, it's more likely that they're going to transition into a similar state than otherwise. And this is exactly the prior knowledge that we encode in something known as the transition plausibility matrix, which in essence is encoding this biological common sense, which is telling you that if you have a kth cell from step I and lth cell from step I plus 1, without doing any kind of optimization, biologically speaking, what can you say about how plausible it is for this transition to happen? And because we are ultimately training a velocity field of a flow matching model using optimal transport couplings, we incorporate this prior knowledge into two kinds of OT novel formulations that we propose. On the left, you have a CTFC formulation which utilizes this transition plausibility matrix and directly transforms the transport cost matrix. Using some simple analysis, you can show that this results in kind of a hard bias as compared to the one on the right, which modifies the entropic term in the OT formulation. And again, using some simple analysis, you can show that this has good mathematical properties in terms of it fitting into existing synchron pipelines, and it also empirically performs better. In terms of data sets, in the main paper, we perform evaluation on 3 data sets, but in the interest of time, I will only talk about 2 here. So the first data set is a axonal post-traumatic brain regeneration data set, or basically a salamander data set. The second one is a mouse embryo organogenesis data set. And how do we evaluate this flow matching model now? Because ultimately we are a generative, because ContextFlow is a generative model, you need to sample from this generative model, right? And what you're learning essentially is a velocity field. Now, if you want to sample the spatial omics data at time t equals 2, and you have observations from time t equals 0 and 1, what you can do is you can start from the known observations at time t equals 1, integrate the velocity field and generate samples at time t equals 2, or you can start at the very beginning and integrate across the horizon and then generate samples at time t equals 2. The latter one is known as the initial value problem sampling, and the first one is the next step sampling. This initial value sampling is a kind of much more harder test of how well you've learned the velocity field across the horizon, because if you make mistakes in middle or in the beginning of this temporal process, you are going to end up with a very divergent generation than what you originally intended. And for the brain regeneration data set, across metrics that are supposed to measure how similar to probability, how similar to sets of samples from some probability distribution are, across those metrics, across sampling methods, across whether you are evaluating the dynamics in interpolation setting or extrapolation setting, ContextFlow performs better over MOTFM, which uses entropic OT couplings and was the prior state of the art, and CFM, which basically uses random coupling. We also evaluate on biological composition, and by biological composition, what I mean is when you start with a certain group of cells and those cells evolve over time, a particular cell type is going to have it's going to divide and it's going to transform into all kinds of different cell types. And at a particular point in time, a particular cell type is going to be present in a particular proportion. So the point of this exercise is how well do you capture those proportions. So you plot the histograms of different proportions of cell types and you calculate the KL divergence. And ContextFlow again has much lower mean KL divergence compared to mini-batch OT flow matching. In conclusion, by the end of this talk, if you understand nothing, you should at least understand that incorporating domain prior knowledge is very much needed in cases where you do not have a lot of data, because the algorithms are performing statistical steps. They do not know the underlying biology. You have to integrate your biological knowledge with the generative modeling framework. And that, in essence, is what ContextFlow is doing. More specifically, we are utilizing tissue microenvironment and cell-cell communication patterns to constrain and bias the generation towards biological feasibility. Going forward, there can be tons of things that can be done. You can think of more innovative ways of incorporating domain knowledge. You can extract mechanistic knowledge and then corroborate with known findings, because at the end of the day, that's what biology is all about. You can use ContextFlow to model spatially aware perturbation responses, or you can even build a spatiotemporal foundation model going in the direction of virtual cell. If you have any ideas for any of these, just let me know and I'll be happy to discuss. Yeah, that's it. Thanks.